Dear student, I am Dr. Prakash Ungli, Professor of Biochemistry. In this lecture, I will be explaining you hemoglobin function. Let's get into the details. Some of the factors that affect binding of oxygen to hemoglobin are the partial pressure of oxygen, meaning the amount of oxygen that is dissolved in the blood is affecting how much oxygen will be loaded onto hemoglobin molecule. And then it depends on the partial pressure of carbon dioxide, meaning the amount of carbon dioxide that is dissolved in the blood will also affect binding of oxygen to the hemoglobin molecule. And also the pH in the surrounding media will influence oxygen binding to hemoglobin molecule. Presence of a molecule called 2,3-bisphosphoglycerate, which is coming from glycolytic intermediate, that is 1,3-bisphosphoglycerate by Rappaport-Leberring shunt, especially in the red blood cell, that will affect release of oxygen from hemoglobin molecule. Temperature within the system will also play a role in oxygen binding to hemoglobin molecule, and presence of any molecules that will compete with the oxygen or same oxygen binding site will influence binding of oxygen to hemoglobin molecule. So, these are all the factors that might affect or may influence oxygen binding to hemoglobin molecule. Now, the effect of partial pressure of oxygen or carbon dioxide. Now, let's look into that one. So, the oxygen dissociation curve that you are seeing down here is a sigmoidal curve. And the sigmoidal curve, it simply indicates binding of very first molecule of oxygen to a hemoglobin, especially to very first heme group present in one of the 4 subunits of a hemoglobin molecule. It takes about 18 millimeter of mercury. So there are 4 binding sites of oxygen in a hemoglobin molecule. Each alpha chain has got 1 heme group, so that means 4 oxygen molecules can bind. So binding of 1 out of those 4 oxygen binding site, so it takes 18 millimeter of mercury as the partial pressure of oxygen, and that is 25% saturation there, because 1 out of 4 has bound with oxygen. Now there is something called as P50, and the P50, as it is shown here, it is the 50% of hemoglobin molecule that is loaded with oxygen, and the P50 for adult hemoglobin, it is 26 millimeter of mercury. That means at 26 millimeter of mercury, 2 out of 4 oxygen binding sites are occupied with oxygen molecule. So note that for binding of 1 oxygen molecule to one of the subunit, it takes 18 millimeter of mercury partial pressure of oxygen. For binding of 2 oxygen molecules to hemoglobin, if we do the simple arithmetic, it should have needed 36 millimeter of mercury, because for 1 oxygen molecule to be loaded, you need 18 millimeter of mercury. For 2 molecule of oxygen to be loaded, by simple arithmetic, it is 36 millimeter of mercury. But in reality, in our body, it takes only 26 millimeter of mercury instead of 36 millimeter of mercury. So there is a difference of 10 millimeter of mercury for loading of the second molecule of oxygen. That means there is something going on here, that is binding of oxygen to one of the 4 subunit lead to a conformational change in that subunit. That conformational change will induce the neighboring subunit to take up a second molecule of oxygen much earlier than otherwise. That's why loading of second molecule of oxygen, it is little more easier than loading of first oxygen molecule, because the first subunit is inducing the second subunit by undergoing a conformational change to take up oxygen molecule. And now once two of these oxygen molecules are loaded onto two subunits. Then those two subunits, as they undergo conformational change, they will induce the neighboring subunit, the third subunit, to take up oxygen. Like that, the fourth subunit to take up oxygen. So when at the partial pressure of oxygen around 80 to 90 millimeter of mercury, almost all four subunits are taken up with the oxygen molecule, means oxygenated. So this type of cooperativity which is going on here. So this is how there is a phenomenon of cooperativity that is going on here in binding of oxygen to hemoglobin molecule, where one subunit is inducing the neighboring subunit to take up oxygen. So that is what is a cooperativity phenomenon that runs in a oligomeric protein like hemoglobin. And because of that, because of the cooperativity phenomenon, you see the sigmoidal curve here. And any molecule or any oligomeric protein that shows a cooperativity phenomenon, its reaction velocity curve or the oxygen dissociation curve here for hemoglobin, it is sigmoidal. Remember that. So usually oligomeric proteins, they will show the cooperativity phenomenon and also they will show the sigmoidal curve in terms of reaction velocity curve or oxygen dissociation curve. Whereas simple proteins like myoglobin do not show a phenomenon of cooperativity. So that's why myoglobin oxygen dissociation curve is hyperbola, as it is shown here. The first curve that you are seeing here, it is a hyperbola, and usually hyperbolic curves have P50 less. That means they will have higher affinity for oxygen. Myoglobin has got higher affinity for oxygen. P50 for myoglobin is 2.8, whereas P50 for hemoglobin is 26 millimeter of mercury, indicating that so hemoglobin has relatively lesser affinity for oxygen than when compared to myoglobin. And also the cooperativity phenomenon that we are talking about, it is also represented by Hill coefficient. Hill coefficient indicates the degree of cooperativity in a oligomeric protein. Higher the cooperativity, lesser the Hill coefficient number. For adult hemoglobin, Hill coefficient number is 2.7. But theoretical maximum of hemoglobin for binding of oxygen is 4. So, as per the Hill coefficient, binding of one molecule of oxygen to one subunit, it gives one number. So, going by that, there are 4 subunits in a hemoglobin molecule. So, the maximum number is 4 there. So, Hill coefficient should be 4 there. But because the degree of cooperativity is going on here, one subunit is inducing another subunit to take up oxygen. That's why you look at the Hill coefficient number, instead of 4, it is 2.7. That means it is decreasing. So, as the Hill coefficient number decreases, the degree of cooperativity is increased. So, Hill coefficient number, remember, it indicates degree of cooperativity. Now, hemoglobin with relatively lesser affinity for oxygen, it is suitable as a transporter of oxygen rather than as a storage form of oxygen. That's why here in the figure you can differentiate. So the P50, if you look at the P50 of myoglobin, which is 2.8, and the P50 of hemoglobin is 26. So going by that, myoglobin has got higher affinity for oxygen and it is used as a storage form of oxygen in our body, whereas hemoglobin with P50 26, it is acting as a transporter of oxygen. Now let's look at effect of partial pressure of carbon dioxide and the pH on oxygen binding to hemoglobin. Now the carbon dioxide from the peripheral tissues in the venous blood is carried to the lungs in 3 ways. As the peripheral tissues are metabolically active, so they will run TCA cycle. So their end product is carbon dioxide, because when metabolic intermediate, that is acetyl-CoA, when it is undergoing TCA cycle, the product of acetyl-CoA is carbon dioxide, one of the products. And that carbon dioxide, it diffuses out of the cell into the extracellular matrix and then appear into the capillaries. So in the capillaries, like where the blood is flowing, red blood cell is there, carbon dioxide is entering into the red blood cell. So inside the red blood cell, carbon dioxide undergoes a reaction catalyzed by carbonic anhydrase enzyme, and further it breaks into bicarbonate and water molecule. Further it breaks into bicarbonate and proton. Now the bicarbonate is pumped out into the blood, and bicarbonate is carried in the blood as the blood returns back to the lung. Whereas the proton that are released in the red blood cell, so it will go and bind with the hemoglobin, because hemoglobin acts as a buffer there, and it is going to protonate hemoglobin molecule. Now the protonation of hemoglobin molecule, it will stabilize a T state of hemoglobin, and the T state of hemoglobin is not a favorable state for oxygen to bind with hemoglobin, so oxygen is unloaded. That's why presence of higher carbon dioxide in the peripheral tissues while entering into the red blood cell, it will create acidity or lower the pH in the red blood cell. By lowering the pH in the red blood cell, so hemoglobin is buffering that proton, and while it does that, so there will be more hydrogen bonds are formed between dimer 1 and dimer 2, and the T state is stabilized. And when T state is stabilized, so it helps in unloading of oxygen. We are going to see that in the subsequent slide. That's what happens to the protons in the red blood cell. And some of the carbon dioxide that is entering into the red blood cell, as it is shown, it will bind with hemoglobins itself and become carbaminohemoglobin, and that carbaminohemoglobin is carried in the red blood cell from peripheral tissues to the lung. So with that, the carbon dioxide that is produced in the peripheral tissues, it can be carried from the peripheral tissues to the lungs in three forms. 80 to 85% is in the form of bicarbonate, as it is written here. Around 15% is bound to a hemoglobin, and that is as a carbaminohemoglobin, and about 5% is dissolved as carbon dioxide. Now let me explain you how oxygen is unloaded in the peripheral tissues and how carbon dioxide is transported from the peripheral tissues to the lungs, and what happens to that in the lungs. So this can be studied by two mechanisms. One is Bohr effect, other is Haldane effect. Now the Bohr effect says it is unloading of oxygen from hemoglobin molecule in the metabolically active peripheral tissues which are having high partial pressure of oxygen and also low pH, meaning increased protons. So as you see here in the figure, in the peripheral tissues, TCA cycle is running at higher rate. That means there is more carbon dioxide being made, which can combine with water to make carbonic acid catalyzed by carbonic anhydrase enzyme. And carbonic acid spontaneously dissociates into bicarbonate and proton, and bicarbonate is pumped out into the blood from the red blood cell. Because all these reactions are going on in the red blood cell, remember that. These reactions are there in the red blood cell. Okay. So the bicarbonate is moving out of red blood cell while chloride is moving in, just to maintain the charge neutrality. And now the protons that are released by the spontaneous dissociation of carbonic acid. So these protons in the red blood cell will protonate histidine residue. And when the histidine residue is protonated, as I said in my previous slide, there will be formation of more hydrogen bonds between dimer 1 and dimer 2. and that will stabilize T state of hemoglobin, which is a deoxy state of hemoglobin. That means binding of protons to histidine, that is protonation of histidine, by stabilizing the T state, it makes oxygen unfavorable to bind with hemoglobin, and so oxygen will be unloaded. So that's how the decrease in the pH, that is increase in the protons in the cytoplasm of red blood cell, will facilitate unloading of oxygen by making more hydrogen bonds between dimer 1 and dimer 2 and stabilizing the T state. Now these protons which are bound to the hemoglobin, that is protonated histidine residues, so as they reach the lungs from the peripheral tissues, when they return to the lungs, in the lungs there is higher partial pressure of oxygen, that is between 90 to 100 millimeter of mercury, and at that high partial pressure of oxygen, so oxygen will be loaded into the oxygen binding sites of hemoglobin. So whatever the number of oxygen binding sites are available, out of 4 maximum binding site, 1 in each subunit. So if all the 4 of them are empty, so 4 oxygen molecules will bind. Otherwise, under normal conditions, in the red blood cell that is coming from the peripheral tissues to the lungs, on an average only 2 oxygen binding sites are empty where oxygen need to be loaded. So at high partial pressure of oxygen in the lungs, so oxygen will be loaded, and when the hemoglobin subunit is oxygenated, it will undergo change in the conformation, and that will lead to breakdown of hydrogen bonds between dimer 1 and dimer 2, and that causes relaxed state of hemoglobin. So in the lungs, R state of hemoglobin is stabilized, whereas in the peripheral tissue, T state of hemoglobin is stabilized. So T state stabilization is unloading of oxygen, R state stabilization is loading of oxygen. So when the oxygen is loaded, when R state is stabilized, means breakage of the hydrogen bonds between dimer and dimer 2. So protons are released because of the breakage of hydrogen bonds, and these protons will go and bind to bicarbonate, which is entering into the red blood cell in the lungs, and this bicarbonate combines with the proton, make carbonic acid, and carbonic anhydrase enzyme breaks it into carbon dioxide and water, and carbon dioxide is exhaled as the oxygen is inhaled. Now we can explain these two concepts, that is the one that is going on in the peripheral tissue and the one that is going on in the lungs. So the mechanism that is going on in the peripheral tissue while unloading of oxygen from hemoglobin in a metabolically active peripheral tissue, it is referred as a Bohr effect. While loading of oxygen and unloading of carbon dioxide from an hemoglobin molecule in the lungs is called as Haldane effect. Okay, so Bohr effect and Haldane effect are due to cooperative interaction between hemoglobin tetramer. Remember that. So it is all about how different subunits cooperate with each other, and based on that, oxygen unloading and oxygen loading will be going on. So these are the things that will be facilitated by the presence of carbon dioxide, lower pH and partial pressure of oxygen accordingly. Now let's move on to see effect of 2,3-bisphosphoglycerate, that is 2,3-BPG on oxygen binding to adult hemoglobin. Now 2,3-BPG, it is synthesized by a glycolytic intermediate called 1,3-bisphosphoglycerate, and that is done in the red blood cell specially by Rappaport laboring shunt, where 1,3-bisphosphoglycerate is diverted into 2,3-bisphosphoglycerate by enzyme bisphosphoglycerate mutase, and thereby there is an increase in the concentration of 2,3-bisphosphoglycerate in the erythrocyte, which is approximately around 5 millimolar concentration. So what is the use of this 2,3-BPG? 2,3-BPG is a anion, it's a negatively charged molecule, 2,3-bisphosphoglycerate. So this 2,3-bisphosphoglycerate will go and bind to some of the positively charged molecules in the interior core of an hemoglobin molecule. So this happens only in the peripheral tissues when partial pressure of oxygen is between 20 to 40 millimeter of mercury. So in the peripheral tissue, 2,3-BPG binds to positively charged pocket between dimer 1 and dimer 2, and it is going to stabilize a T state of hemoglobin, and that helps in unloading of oxygen from hemoglobin. That means presence of higher 2,3-BPG, especially in the peripheral tissues, will help in unloading of oxygen from hemoglobin. Whereas when the red blood cell reaches lungs, even if 2,3-BPG is available in the red blood cell, so while red blood cell is there in the lungs, when partial pressure of oxygen is 90 to 100 millimeter of mercury, so oxygen will still be loaded onto our hemoglobin, that is heme subunit in the hemoglobin, and our state is stabilized there. So 2,3-BPG do not bind to hemoglobin in the lungs, because in lungs our state is stabilized by higher partial pressure of oxygen. Now the other thing is 2,3-BPG do not interact with the fetal hemoglobin. That's because the positive pocket that you see in adult hemoglobin is present between beta 1 and beta 2 subunit. That's where 2,3-BPG will interact. Whereas in fetal hemoglobin, the subunit combination is alpha 2 gamma 2. That means we have gamma here instead of beta in adult hemoglobin. So gamma subunits do not have that positively charged pocket to interact with 2,3-BPG. That's why 2,3-BPG do not interact with fetal hemoglobin, and that will lead to fetal hemoglobin having little higher affinity for oxygen than adult hemoglobin. So the P50 for fetal hemoglobin, it is 20 millimeter of mercury compared to P50 for HbA, which is 20 millimeter of mercury. That's the difference there. So the difference of 6 millimeter of mercury, because of not binding to 2,3-BPG by fetal hemoglobin, it makes that fetal hemoglobin having higher affinity for oxygen than adult hemoglobin. And that is a survival advantage for the fetus, because fetus has to get oxygen from the maternal side. Having HbF in the fetal circulation, it is helpful in getting oxygen from the maternal blood, which has HbA, across the placental membrane. That exchange has to go on, and that gaseous exchange is possible only because fetal hemoglobin is relatively having higher affinity for oxygen compared to adult hemoglobin, and that comes with the difference in binding of 2,3-BPG between fetal hemoglobin and adult hemoglobin. So here is how effect of 2,3-BPG binding to hemoglobin is shown, and thereby altering its oxygen binding capacity. As you see here, between beta 1 and beta 2, there are positive charge pocket, especially in the interior core, and 2,3-BPG is a negatively charged molecule, so it's going to interact with these positively charged amino acids like lysine and arginine, and thereby it makes a connection between B1 and B2, that is dimer 1 and dimer 2, and it brings this dimer 1 and dimer 2 together, and thereby it is going to cause T state stabilization, and oxygen binding to T state of hemoglobin is unfavorable. So oxygen binding is unfavorable here. That's why hemoglobin will release oxygen. That is why 2,3-BPG helps in unloading of oxygen from hemoglobin molecule. So what is the clinical significance of 2,3-BPG? So whenever there is an increase in the concentration of 2,3-BPG, it leads to unloading of oxygen more readily than when 2,3-BPG is deficient. So concentration of 2,3-BPG in the red blood cell, it will be in response to various physiological and pathological conditions. Like there will be increase in 2,3-BPG in conditions of oxygen deprivation, like when less oxygen is available for loading process. So whatever the oxygen is loaded, like out of 4 subunits, if only 2 subunits or 3 subunits are loaded with oxygen in the lungs, if the red blood cell containing hemoglobin, which is partially loaded with oxygen, if it is coming to the peripheral tissue for proper oxygenation of the peripheral tissue, so the efficient unloading has to go on, because already hemoglobin is bringing less oxygen to the peripheral tissue. So the efficient unloading of oxygen in the peripheral tissue, it can be facilitated by having increase in 2,3-BPG. That's why in conditions of oxygen deprivation like lung diseases, chronic anemia, or during adaptation to the high altitude, there will be always increase in 2,3-BPG. But also note that when the blood is stored for a prolonged period of time, as the glycolysis is kind of continued, right? So there will be decrease in 2,3-BPG content, because 2,3-BPG will be converted into 3-phosphoglycerate, and 3-phosphoglycerate will be converted into 2-phosphoglycerate, and 2-phosphoglycerate forming phosphoenol pyruvate, and that eventually form pyruvate. So that means storing of blood for a long period of time as the glycolysis continues in the red blood cell. So there is a continued use of 2,3-BPG, putting that into glycolysis, so the levels of 2,3-BPG content in the stored blood will decrease. So how to avoid this decrease in 2,3-BPG content? And that is done by adding inosine into a storage media, because inosine is going to prevent decrease in 2,3-BPG, as inosine gets into pentose phosphate pathway and become a ribose 5-phosphate, and that ribose 5-phosphate can get into a glycolysis in the form of fructose 6-phosphate and glyceraldehyde 3-phosphate, and continue to provide glycolytic needs and also decrease the conversion of 2,3-bisphosphoglycerate into 3-phosphoglycerate. So this is how a depletion of 2,3-BPG in the stored blood can be regulated or prevented. So the central curve that you are seeing here is the normal one, where you see this sigmoidal curve, with all that normal conditions, normal 2,3-BPG, normal temperature, normal PCO2, normal PO2, all that. That's what we get. And the P50 is 26 millimeter of mercury. Now, whenever there is an increase in PCO2, whenever there is increase in temperature, whenever there is a decrease in the pH, meaning increase in protons, and whenever there is a hemolytic anemias going on, so in all those conditions, or whenever there is increase in 2,3-BPG, like physiological adaptation to having lack of oxygen, chronic lung diseases, in all these conditions, living in the higher altitude, that includes, so in all these conditions, there will be efficient unloading of oxygen from hemoglobin molecule, and that will shift oxygen dissociation curve towards the right side. So right shift in the oxygen dissociation curve is T-state stabilization and release of oxygen from hemoglobin, because these factors directly or indirectly is going to decrease affinity of oxygen for hemoglobin, because whenever there is increase in carbon dioxide, PCO2, carbon dioxide will combine with water to make carbonic acid, and carbonic acid breaks into bicarbonate and proton, and that decreases the pH in the red blood cell, and those protons will go and protonate histidine residue, increase the positive charge there, and that protonated histidine will make more hydrogen bonds between dimer 1 and dimer 2, stabilize the T state and unload the oxygen. So oxygen is unloaded. Like this, every factor that are given here, like increase in PCO2, decrease in the pH, increase in the temperature, increase in DPG. DPG means previously DPG is now it is written as 2,3-BPG. 2,3-BPG is called as DPG. DPG for diphosphoglycerate. Diphosphoglycerate means 2,3-biphosphoglycerate. Bi means 2. Di or bi, it is 2. Okay, so increase in 2,3-BPG and also anemias, different kinds of anemias. It can be sickle cell anemia, it can be thalassemia, it can be any kind of anemia, or living in high altitude, or chronic lung diseases. In all these conditions, whenever there is rise in 2,3-BPG, so it is going to shift the oxygen dissociation curve towards the right side, and that is decrease in the affinity of oxygen for hemoglobin, because all these factors will stabilize T state of hemoglobin. Okay, so at that time what will happen to the P50? P50, that is of hemoglobin, will rise. So P50, there will be increase in P50 of hemoglobin, because affinity of oxygen for hemoglobin molecule is decreased. It takes more partial pressure of oxygen to load oxygen onto hemoglobin molecule in the presence of increased levels of these factors. Now let's look at the opposite condition. So whenever there is left shift in the oxygen dissociation curve, let me take this out and show you the left shift now. So when there is a left shift in the oxygen dissociation curve, as you see, the P50 will decrease. So the decrease in P50 means there is increase in the affinity of oxygen for hemoglobin molecule, and any factor that decrease P50 and shift the oxygen dissociation curve towards the left side, they will stabilize R state. R state stabilization means oxygen is not easily released from hemoglobin molecule. So oxygen binds with this hemoglobin with higher affinity. And that means some of the factors that were unloading oxygen previously, helping in release of oxygen from hemoglobin molecules, now those factors are decreased. That's why oxygen dissociation curve is shifted to the left side, where oxygen is binding to hemoglobin with little more higher affinity. So those factors can be decrease in partial pressure of oxygen, decrease in the 2,3-BPG content, decrease in the temperature, increase in the pH, methemoglobinemia, where hemoglobin ferrous is converted to ferric, carboxyhemoglobin, fetal hemoglobin, basically hemoglobins having higher affinity for oxygen, this type of things are increased, right? So they all will shift oxygen dissociation curve towards the left side, making affinity of oxygen for hemoglobin much higher, thereby it is not readily released from the hemoglobin. So those are the factors that I have written here. Just as a recap, let's look at them. So the factor shifting oxygen dissociation curve towards the right is living in high altitude, which increases 2,3-BPG, chronic restrictive or obstructive lung diseases, which will increase 2,3-BPG, shifting the oxygen dissociation curve towards the right side, because it will stabilize the T state and help in unloading of oxygen. Chronic hemolytic anemias like sickle cell anemia, thalassemia, all of them increase 2,3-BPG, thereby shifting oxygen dissociation curve to the right side. Increase in the tissue temperature like exercising skeletal muscle will help in unloading of oxygen. Increased tissue metabolism, which will lead to increase in PCO2, increased H+ ion concentration, meaning decrease in pH, will help in unloading of oxygen from hemoglobin. And abnormal increase in organic acids, like leading to metabolic acidosis, will help in unloading of oxygen from hemoglobin molecule. And when the oxygen is unloaded, so when the hemoglobin is returning back, it will bring carbon dioxide back to the lungs, and carbon dioxide is exhaled and more oxygen is loaded, right? So that will increase the respiratory rate and it will lead to hyperventilation in metabolic acidosis. Remember that. Now let's move on to see conditions that will cause left shift in the oxygen dissociation curve, and that is respiratory alkalosis, where there is decreased PCO2, H+ ion concentration, So thereby there is stabilization of R state of hemoglobin, and R state hemoglobin, it will hold on to oxygen with much higher affinity. And hypothermia, decrease in 2,3-BPG, where if there is a mutation in 2,3-bisphosphoglycerate mutase enzyme, or mutation in beta chain of amino terminal amino acid, where it alters the positively charged pocket in the beta chain where 2,3-BPG is binding. If that kind of mutation is there, where positively charged amino acids are replaced by some other amino acids like neutral or negatively charged amino acids, so 2,3-BPG may not interact properly, so it will behave like a gamma chain in terms of binding up 2,3-BPG. That will also increase affinity of oxygen for hemoglobin because of lack of binding of 2,3-BPG, and the shift oxygen dissociation curve towards the left side. And also presence of carbon monoxide. Carbon monoxide, it increases affinity of oxygen for hemoglobin molecule by relaxing the chains, and thereby it will increase affinity and shift in the oxygen dissociation curve towards the left side. And also presence of HbF in the red blood cells. HbF, instead of beta chain, there is a gamma chain, and gamma chain do not have positive charge pocket to bind with 2,3-BPG. So absence of 2,3-BPG binding will make fetal hemoglobin having higher affinity for oxygen, and that's why it will shift oxygen dissociation curve towards the left side. So these are the factors that will affect binding of oxygen to hemoglobin and shift oxygen dissociation curve either to the right side or to the left side. So this is very important concept, especially for different kinds of exams, and also you should have an idea like how exactly oxygen is loaded onto hemoglobin molecule, what is cooperativity phenomenon, what is Yield coefficient, how many molecules of oxygen bound to hemoglobin, and how each subunits will cooperate with one another, what is P50, and how carbon dioxide is carried from the peripheral tissues to the lungs, and what is Bohr effect, what is Haldane effect, and what is the clinical significance of 2,3-BPG. So all these details are most important. I hope this lecture has helped you in understanding this important concept in biochemistry. Thank you.